Okay, here we go. Let's talk more immunology. Chapter 3. And we'll stop that from jumping ahead. There we go. So, yeah, this chapter is about pattern recognition. So, pattern recognition is going to be one of the parts of the innate immune system. And we're going to look at things like NOD proteins and toll like receptor proteins, among others. And part of what we're going to memorize for this is you're going to be able to tell me all of the ligands, so the things that toll-like receptors bind. And the word for that is ligand, is just a thing that binds to a receptor of some sort. And so, yeah, here we go. And you're also going to tell me what they interact with, which is going to be, I think, one of two proteins. But yeah, we can get this. Okay, so, and then we'll also talk about what these toll-like receptors do and a little bit about where they came from. And yeah, the nod-like receptors are similar. They're not as big a deal in humans, but in animal evolution, these are a very prominent thing. Okay, so jumping ahead, here's our introduction. And this is nice. This kind of shows you where we are. So, innate immunity. Yeah, these are things that are only inherited through the genome. So the specificity is determined by the gene. In other words, the gene can only do one thing, and it either binds that thing or doesn't bind that thing. And so this is how the immune system of most animals works. They will have these what are called pattern recognition receptors, which are just able to pick up really basic generic forms of danger. So things that would be present in any bacterial cell wall, for example, or things that would be present in a fungus, for example, or things that would come out of just an average virus without knowing which one it was. These are the sorts of receptors, and so they can be broadly useful, but they're not as specific as you might like. So it's difficult to tailor these to a specific type of response. You pretty much make immune response or not immune response. Those are your choices. Yeah. Some of these innate markers are expressed on most of the cells of a particular type, but variable here actually means no. They just didn't want to put no. And innate immunity, these things will have a very quick, you could say, immediate response. And yeah, they recognize broad classes of things. Like, for example, lipopolysaccharide is one of the components of a bacterial cell wall. So lipopolysaccharide is LPS, that's how we abbreviate it, is going to be one of the things that is triggered by these pattern recognition receptors, or rather one of the things that triggers them. So yeah, it's going to interact with a range of molecules, even though they're slightly different. It's still going to be able to bind them within limits. If a bacterium comes up with a new way to modify this molecule, it may not bind to the pattern recognition receptor that used to bind it. So take that yes with a grain of salt there. Okay, adaptive immunity is all about things like antibodies and things like T cell receptors. These are little libraries of genes that are like our box of Legos. We're going to pick out different parts and put them together and build a molecule. So we've got to have a top part, we've got to have a middle part, we've got to have a joining part, and we've got to have a bottom part every time. But there is a library of choices for each one of these, and we can then go in and modify each one of those a little bit further to try and find new ways to specifically interact with a disease. So these guys up at the top, these innate immune receptors, these are things that you would actually find in some form in every kind of animal. And I don't just mean vertebrate animal, which are kind of things with bones like you would see in the zoo, but also in insects and something like a jellyfish and just, worms of different kinds will have innate immune systems of this sort. Adaptive immunity, as we understand it, seems to be a thing that you only find in vertebrate animals. So, in things that do have bones, so basically everything from fish all the way up to, various mammals and other lizards and things like that. Yeah, so those are, encoded in bits and require a little bit of assembly slash rearrangement. And then once they make a rearranged version of an antibody or a T cell receptor, then you'll get lots of copies of that cell being formed, um, carrying the brand new molecular signature, whatever it is. And yeah, some of these things are able to distinguish between even closely related structures, which you can't be both yes here and yes over here interacting with a broad range. I mean, right? Those two seem like they're contradictory at some level. And so that's the issue, I think, with this table. There's a bit of both in here, yes, but neither one of these is 100% true. All right. So, what's the big idea? Well, all right. You've got these phagocytes, which are going to be things like macrophages, and neutrophils can do this to some extent. And they are going to have a variety of receptors on them. So, a macrophage's job in this world is to latch on to something and pull it inside and eat it. Now, macrophages don't just eat everything. Otherwise, they would consume red blood cells and all the proteins in our, bloodstream and just start, tearing into cells that we need. Macrophages are going to be triggered in their eating response when they get a signal to one of these, what we call pattern recognition receptors. So, for example, things like scavenger receptors or lipid receptors or FC, this is binding to the bottom of an antibody, or complement receptors. So we learned about complement the other day. And yeah, one of the functions of complement is to make it easier for a macrophage to eat. And it becomes easier because the complement is going to be like a handle. It's going to be stuck to the bacterium just like that. And since we have complement receptors on the macrophage, boom, as soon as those receptors engage a target, they're going to pull it in and digest it, just like so. Yeah. And so, yeah, also these dectin and mannose receptors, those would also be active, perhaps more against a fungus than bacteria, for example. But you can put complement onto pretty much anything that's invading the body. And so a complement receptor is already a pretty broad sort of thing. And then an FC receptor just says anything that you made an antibody to is worth killing. Worth killing and digesting. That's how macrophages see the world. It's a very simplistic worldview from this perspective. And so the way that this is done is that they just latch onto these things, they bring them inside the cell where the bacterium or virus or yeast or whatever cannot escape, and then they're going to merge that little compartment. So they're going to bring it inside of a little bubble called a vesicle, or called a phagosome, because this is how we're going to eat. And then we're going to merge that with a thing called a lysosome, which has a lot of enzymes in it that will break down protein or sugar or lipid. And it also has very low pH, which helps to break up RNA and DNA. And so, yeah, we're essentially going to shred this thing down to nutrients and then absorb them. So the macrophage is theoretically eating and gaining, nutrition, basically, from the things that it's fighting to protect us. So that's a nice little thing. It's like, a car that runs on garbage, like in Back to the Future 2 or 3 or whichever one that was. Okay, so G protein coupled receptors. All right, this is a very wide, common form of receptor. This is how most of the signaling is done in your cells. So when a cell is going to respond to the environment, most likely it happens through a thing called a G protein coupled receptor, or GPCR. And these are things that we cover in Bio 1, just because I like hammering on them because they're so important in biology. There was a statistic that something like greater than 90% of all FDA-approved drugs are actually targeting GPCRs. So all those things that change your mood, most things that are involved in like anti-allergy pills of some sort, these are all little molecules that bind to a G protein coupled receptor and either stop it from working or make it work when it wouldn't have worked otherwise. That's the idea. So essentially, yeah, you're going to have a ligand coming along up here, and the ligand is going to bind to the G protein coupled receptor. when it binds, it actually causes a change in the shape of the receptor. So instead of opening up like this, it's got 7 times it goes through the membrane. Number 5 and a little bit of the ones next to it actually get pulled into the center. So it does have this sort of shape, but it doesn't really spread out the way this is doing it. But when it does that, the G protein coupled receptor is then able to bind to this thing, which is called a G protein. And there are different versions of G proteins. Some have 2 parts and some have 3 parts. And so they'll be labeled alpha, beta, and gamma, or just alpha and beta for the different parts of the G protein. And these things are going to be anchored in the membrane, and they are going to break apart. They're going to split off. So alpha will go to a different place than beta and gamma once the GPCR is activated. It's basically kicking this protein in such a way that it breaks into 2 pieces. And the 2 pieces then go off and activate other proteins and cause other effects. And so as part of the activation, we start out with a guanine, like the nucleotide, GTP. And in the inactive form of the protein, which is how it starts out, that gets knocked down to a GDP. So it's guanine diphosphate. It just has 2 phosphate groups on it instead of 3. And so the first thing that happens once it gets free, just like a person coming out of prison, it just wants to get rid of this old, used up G protein and pick up a new one. So that's what it does. And then boom, 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 triphosphate. Now this is the active version. The active version can interact with different proteins. And it'll interact with a bunch of different proteins that we're going to cover a little bit later. But the alpha subunit and the beta and gamma subunits have different effects on different proteins. And then when it's finally done, then the alpha subunit is going to very slowly cut off the 3rd phosphate. And so it's going to go from GTP back to GDP, and that's how it turns off. So it's like a timer. It's like you set the timer for 15 minutes of furious activity, at the end of which this protein is going to turn itself off, which is very nice. It's like self-regulating. And then once it is turned off, it can now bind again to the beta and gamma subunits here. And these things will only stick to the alpha subunit when it is the GDP form. As soon as it becomes the GTP form, it's actually going to break these apart. So as it binds this is moving away. It's like one or the other, All right. So what can G protein coupled receptors on phagocytes, things that cells that eat other material, including other cells, what can they do? Wow, so many things, right? So we're focusing on macrophages here and neutrophils, but among the various things that they can do. So both macrophages and neutrophils can acidify. So they're acidifying internal compartments, like those lysosomes. And normally these things would start at pH 7 and would drop down toward pH 5 or so before they get merged with the lysosome. But the cells are able to turn this process up and get the pH all the way down to a very acidic 3.5 to 4. I believe straight up vinegar is somewhere around 4.5. So this is, really acidic. And that acid will either break down bacteria or it just makes life so difficult that they cannot continue to divide. So bactericidal, these are 2 good words to know. Bactericidal means you've actually killed the bacterium. Like homicide, bacteria, homicide, boom, smush them together, bactericide, Bacteriostatic is a different thing. So this is just making the world a little bit difficult for the bacterium so that it cannot continue to grow and divide. So the number of bacteria is supposed to stay pretty much the same, or they'll grow at a very slow rate compared to normal. Either way, this buys time for the rest of the immune system to either come in and clean up the problem if you're being bacteriostatic, or to clean up the mess after the problem if you've gone bactericidal. Okay. Among the other things that your immune system and macrophages and neutrophils in particular can make is hydrogen peroxide. That stuff that can bleach your hair, and some people brush their teeth with it or do a mouthwash with it. What it does is it breaks down into free radicals, and you've probably heard of these things. This is, a hydroxyl molecule right over here that has one extra electron but is short one electron, so it doesn't quite have a complete set. And this little thing will react with whatever the next molecule is that it comes into contact with. So the H2O2 is like a hand grenade, and when you split it open into the two OH radicals, hydroxyl radicals, then each of those is now basically ready to detonate. It's going to break something. So these cells will secrete H2O2 and hope that the H2O2 does more damage to the bacteria or fungus or virus or whatever than it does to the cell that just produced it. Yeah. It's not a, super safe way to do things, but it's effective, and it is widely done by macrophages and neutrophils. Another way to do this is with nitric oxide, and same sort of deal. It is a toxic nitrogen and oxygen compound that can be made by both of these cells. These antimicrobial peptides are things that we've mentioned earlier, so things like defensins and cathelicidin and all these other related proteins. Will have direct antimicrobial, so mostly antibacteria properties. They can just latch onto them and punch holes into them until their guts run out, basically. That's something I think we covered in chapter 1. Okay, these phagocytes can also produce lysozyme, and again, that's both macrophages and neutrophils will do this. And so that is something that is going to break down a bacterial cell wall, which I think we mentioned earlier. And some hydrolases that will just break down some of the pieces that are left over from these ingested microbes. So just because a macrophage or neutrophil has eaten a bacterium doesn't mean it can necessarily just automatically break that thing down. Sometimes it takes some work. And a lot of these enzymes that are produced are going to be produced in response to G protein coupled receptor signaling. All right. And just the last little bit on this section before we pause this particular part. Let's see. The point of the G protein coupled receptors is they're going to link up different processes. So macrophages and neutrophils have the ability to destroy things that they ingest, and they have the ability to ingest things. But you don't want them just ingesting everything. And so what the GPCRs do is they're going to link these processes together. So when there is danger, then you want your neutrophils and macrophages to go out and start eating things, breaking them down. Yeah, even if it's non-discriminate, even if you lose a couple of your cells, it's going to be worth it in the long run. So, for example, one of these things is called all mammalian proteins start with an amino acid called methionine. And so we abbreviate that as MET. Bacterial proteins don't start with methionine. They start with a thing called formal methionine. It has a formate molecule on it. So that's this FMET, FORMYL, methionine. And a lot of proteins actually start with a formal methionine, then leucine, then phenylalanine, those 3 amino acids in a row. Those 3 together, if they are present, will actually set off GPCR that's going to signal to a protein called RAC2. And then RAC2 will cause assembly of this sort of killing chamber where we're going to release different molecules. We're going to bring in a lysosome and a thing called a secondary granule and a thing called a primary granule. The primary granule is going to have enzymes inside of it. The secondary granule is going to have those reactive oxygen species. The lysosome is going to have peroxide. And the OCl minus is the active ingredient in bleach. And so, this is also something that cells in your immune system can make. And you know what? It works on your countertop, and it works inside your cells to destroy bacteria. And SOD is a superoxide dismutase. That's another thing that can be used to attack bacteria. And there are a bunch of other enzymes that would also be involved. But this is the process. So RAC2 is going to cause all 3 of these little vesicles, all 3 of these little pods to come together and merge with whatever has been brought in the brand new phagosome. And so, yeah, that's how bacteria get destroyed. And so here you can see it actually happening. So you've got your white blood cells down here. So these are going to be something like a neutrophil or a macrophage, these yellow guys. You've got your little pink cells here. These are going to be bacteria. Something like E. coli would have this particular shape. And then you see all the blue stuff covering those. This is the neutrophil extracellular trap, or the net. This is what neutrophils can do. They rip themselves open, and this is literally the sticky DNA of neutrophils that have destroyed themselves in order to fight this bacterium. And you can see, look at all of the bacteria that have got stuck to the net. It's just like a spider web. And it's going to catch these bacteria, and even if they start to divide in there, like these guys are trying to do, it's okay, because the mama bacterium and baby bacterium, which are really just two halves of the same thing, are both going to be trapped. So yeah, nets, neutrophil extracellular traps, are also very useful part of the immune system. And these are going to be activated when the neutrophil itself is activated. And those G protein coupled receptors are one way that neutrophils do get activated. Okay, so let's pause this section and then we'll go on to the next section in the next video.